Exam 1 week, countdown to weekend 1 hour. So you can get all kinds of studying done in preparation for the exam. I will schedule a review session. I have not yet decided on when that will be. That will most likely be either Tuesday evening or Wednesday evening. And I will announce that likely on Monday. So you'll know that. Exam will likely cover through all of this. And it might finish just a little bit before there. I haven't decided, but it'll likely go all the way through all this. Okay, so last time I got started talking about the DNA polymerase and how DNA polymerases work. I want to say more about that today and also about other proteins that are involved in the replication of DNA. So the thing I talked about last time very briefly was the elongation process, and in the elongation process, a 5' phosphate of an incoming nucleotide is covalently joined to the 3' hydroxyl of an existing nucleotide, which is why there has to be an existing primer in order for DNA polymerase to work. And that creates a phosphodiester bond, and then the polymerase scoots down one more nucleotide and does the same thing. Along the way, it checks to make sure that it has properly base paired those, A going with T, G going with C, for example. And I pointed out the very astonishing feature that this is occurring at the rate of 1,000 nucleotides a second with at least one of the polymerases in E. coli, with an error rate of in E. coli of about 1 in 10 million, which is a really remarkable feet. In eukaryotic cells, the error rate is roughly 1 in a billion. So it's pretty incredible how the polymerase is able to do all of this with such incredible speed. And we'll see more about speed as we work through things today. One of the ways in which DNA polymerase is actually so good at doing what it does is that not only is it working at a net rate, this is not an after effect rate, but a net average rate of about 1,000 nucleotides per second, but it has a function, or at least most DNA polymerases have a function built into them called proofreading. So just like you would type a document and before you turned it into your professor, I would hope that you proofread it, and that meant more than just running it through a spell checker, but in fact that you actually looked at it and searched for any errors or embarrassing errors that were there. Proofreading is an interesting phenomenon, and as I say, it's not found in all DNA polymerases. So DNA polymerases that lack proofreading ability tend to be what we call error-prone. So there are numerous examples of error-prone DNA polymerases, and many of these are found in viruses that code for their own polymerase. HIV virus is a very good example. It has a DNA polymerase called reverse transcriptase that lacks proofreading. And so consequently, instead of having an error rate of about 1 in many million, it has an error rate of about 1 in 10,000. And while that's still better than you can type, it means that for a virus that replicates many, many times, it's going to accumulate many, many errors relative to the number of errors a cellular polymerase would create. That's important for a virus like HIV, which evolves ways around drug resistance and ways around cellular protections very rapidly because more mutation actually favors that. Mutation tends to work against a cell because a cell has certain things that it needs to maintain being alive, whereas a virus is pretty much, let's see if this works or not. And that's how viruses work. And so that error-prone replication of viral DNA polymerases is a very, not an uncommon component. Well, let's talk about how proofreading works. Proofreading is a very interesting phenomenon. I want to come back to thinking about the elongation process for DNA polymerase. So DNA polymerase moves along, it grabs a hold of an existing strand, and it starts at the 3' end and moves in the 3' direction. So it's actually going 5' to 3', adding things to the end of the chain. If we think about the DNA polymerase going along like this, And it sees an A and it puts in a T, and it sees a G and puts in a C, and sees another C and puts in a G, and sees another C and puts in a G, and sees a T and puts in an A, and sees a T and puts in a C and goes, uh-oh, right? A C doesn't pair with T, right? So everything I described up until that point was perfectly paired. And so the polymerase is just scooting along fine, but it gets up here and it has made a mistake. Just like you have mistyped a key on the keyboard of your computer, so too has the polymerase grabbed the wrong nucleotide and it's put a C across from a T. Okay? Well, ideally, the polymerase will recognize that and fix that error. And so one of the ways in which a polymerase does that is actually by the phenomenon of proofreading. So if it's going along doing fine and all of a sudden it encounters an error, it stops. Okay? Well, as it was moving, putting the things in, it was moving in the 5' to 3' direction, and it's what we call a 5' to 3' polymerase activity. That's what it's doing as it's making the strand, a 5' to 3' polymerase activity. But it gets up here to this error, and it stops. Well, it's already made the bond between the previous nucleotide and that misdone C. So it's already linked in there. It's got to get rid of that C. How does it get rid of that C? Well, that means it's got to break that bond between the C and the previous nucleotide. And to do that, it has an activity, and this activity is found in the polymerase. It has an activity called a 3' to 5' exonuclease. What does that mean? Well, it was moving 5' to 3' this way. 3' to 5' would mean it's got to back up, right? And that's what it does. It backs up, and it clips that phosphodiester bond. That's the exonuclease. Because an exonuclease starts at the end of a DNA strand and starts chopping out nucleotides by breaking phosphodiester bonds. So that's what the proofreading is doing. 5' to 3' polymerase for making DNA, 3' to 5' exonuclease going backwards to remove errors. Okay? Well, it turns out that that 3' to 5' exonuclease improves the accuracy of a DNA polymerase by about 10 to 1,000-fold. Depends on the polymerase, but it's a significant improvement in the accuracy of that polymerase. Now, I want to emphasize that the polymerase is not perfect. Even if you proofread something that you write for your professor many times, or through your letter to somebody, and then as soon as you send it, you go, God damn, what an embarrassing error, right? So even though you proofread things, errors can make it through, and the same is true for DNA polymerase. The proofreading reduces the number of errors, but it does not completely eliminate them. Okay? So those numbers that I quoted for you of 10 million, 1 error in 10 million to 1 in a billion, okay, are after the proofreading has happened. If we don't have proofreading, we get about 10 to 1,000 times more errors depending upon the polymerase. Okay, so that's how proofreading works. When we think about the replication of DNA, however, in the cell, there's a lot more to it than DNA polymerase moving along and having nucleotides. So far what I've talked about is that for replication to occur, you have to have a strand of DNA that has another strand paired to it that can be elongated, so you have to have that. I have to have a polymerase, and I have to have 4 DNTPs. And if I do that in a test tube, I will get replication of DNA. It won't occur at the rate of 1,000 nucleotides a second, and it won't be as efficient as the cell will do it, but it will replicate it. When we look in the cell, we discover that the replication process has something we call replication machinery. Excuse me. Because like a little machine, there are several proteins at the place of replication where they have specialized functions that they perform. The site of replication is called the replication fork. I may have used that term last time. A replication fork is the place where replication is occurring. So what I want to do is go to a replication fork that shows you some of these proteins and then talk about their individual functions. So I've gone from talking about DNA replication in a test tube to talking about DNA replication now as it occurs in a cell. Okay? Talking about DNA replication as it occurs in a cell. And if we look at a replication fork, it looks something like this. And that's different from that very simple Y-shaped structure I showed you the other day. There is a Y. It's just not quite as exaggerated. There's one strand of the Y, and there's the other strand of the Y. And we discover some interesting things that are here. We remember, first of all, that replication can only go 5' to 3', which means if we're looking at the lagging strand, we've actually got to loop this thing around backwards in order to make that strand be oriented in the 5' to 3' direction, or the 3' to 5' direction, so we can copy it 5' to 3'. By the way, don't forget that. A strand that's working 5' to 3' is reading the other strand in the 3' to 5' direction, right? Because they're anti-parallel, right? Okay. Well, let's take a look at this. If we look at it, we see at the point of action, we see a DNA polymerase. And that DNA polymerase in E. coli has a specific name. That specific name in E. coli is it's called DNA polymerase 3. Okay? DNA polymerase 3 is the polymerase in E. coli that replicates the vast majority of the cellular genome, or of the cellular genome, okay? The genome of E. coli is about 6 million base pairs in length. It is a circle, okay? Bacterial DNAs are circular, okay? Eukaryotic DNAs, like we have in our cells, are linear. And that's a very fundamental difference that we'll talk about later, okay? So the DNA of a bacterium is circular. And that actually provides some advantage and some simplicity, as we will see, for the bacterium. But we're not concerned about that for the moment. We're concerned about this DNA polymerase, okay? This DNA polymerase is actually, it's what we call a dimer, meaning two identical pieces. And one piece is replicating the leading strand, and the other part is replicating the lagging strand. And the leading strand part can actually get ahead of the lagging strand part. Not surprising, that lagging strand part has got to wait, has got to try to catch up all the time, and that leading strand can get ahead because it doesn't have to wait for primers and things like that. So if this leading strand gets ahead and the lagging strand is behind, what we can see is that this strand that's been pulled out can actually be a piece of it sitting there waiting to be replicated. And it loops out and forms what we call a trombone structure. Like a trombone moves in and out, this looping piece can go in and out as well, depending upon how fast the lagging strand synthesis is proceeding. Well, this polymerase 3 has a 5' to 3' DNA polymerase activity. It has a 3' to 5' proofreading activity. But this polymerase, if it were left on its own, would not work very well. And the reason it wouldn't is it really ideally needs to have these strands separated. It really ideally has, not ideally, but it absolutely has to wait for the primer to be made, okay? And it turns out that when we start separating strands, we create problems like knots. And we don't want knots to form in the DNA. So fortunately, the cell has all kinds of proteins to deal with that. Now, I said I'm talking here about E. coli. What happens in eukaryotic cells, like our cells, is very similar. Very similar. Okay? The proteins have different names, and it is a bit more complex. But for our purposes, we will talk about it occurring in the same general fashion. And for the most part, I'm not going to talk about different names of eukaryotic proteins. I'll give you a couple, but for the most part, I won't. So we'll treat it all DNA replication, unless I note otherwise, in the same way. Okay? Well, what does this need? Well, first thing I said it needs to do is the cell needs to peel these strands apart. Because if the DNA polymerase has to peel the strands apart, it has to take time to do that. It has to take energy to do that, and that slows everything down. And the cell, an E. coli cell with 6 million base pairs, and it turns out a pair of these DNA polymerase is working needs to be working at 1,000 nucleotides a second because it needs, under ideal conditions, to be able to replicate the entire genome in about 20 to 25 minutes. Okay? So this has got to have an optimized system for replicating DNA. Well, let's talk about separating the strands. Separating the strands is the first thing I usually talk about, and it's fascinating. This protein up here, called helicase, okay, is an incredible protein. Its job is to peel apart those strands. Duh. Well, peel apart the strands. Well, you suppose that's going to take energy? Yep, it's going to take ATP energy. Helicase requires ATP energy. How fast does the helicase have to peel apart the strands? Well, it needs to stay ahead of the DNA polymerase so they don't bump into each other. And that means that the helicase needs to be working at about 1,000 nucleotides a second. Okay, so what? Well, here's what, so what. 1,000 nucleotides a second, there are roughly 10 nucleotides, or 10 base pairs per turn, right? That means that it's doing 1,000 nucleotides a second. It's therefore doing 100 turns per second. And if you take 100 turns per second and you multiply it times 60 seconds per minute, you discover that helicase is operating at 6,000 RPM. That's remarkable. Your car is not operating at 6,000 RPM unless you're, shall we say, driving beyond the speed limit significantly. Okay? Now, this guy does it routinely, zipping through that DNA. When we talk about machinery, there's a reason we talk about machinery, because this thing is acting like a motor. It's acting like a motor, 6,000 RPM to stay ahead of that thing. Well, let's imagine what's happening with this helicase. Imagine this helicase is here, and it's peeling apart these strands. And we see these strands pulling out up here. What do you suppose is happening ahead of the helicase? Do you suppose that things are going to get very tight? Do you suppose they might tend to form a knot? The answer to both of those is yes. Okay? Both of those will happen unless the cell does something to prevent that. Well, one of the things that the cell does to prevent that is that the cell has an enzyme, and by the way, helicase is an enzyme. All these various things, or most of these things I'll talk about, are enzymes. Helicase has an enzyme, not helicase. The cell has an enzyme ahead, and this makes it look like it's right next to the helicase. It's not. It's actually a little further up away from there. Okay? This enzyme is something we call a topoisomerase. Topo, T-O-P-O-I-S-O-M-E-R-A-S-E, topoisomerase. And topoisomerase's job in the cell is to prevent knot formation by releasing tension. Okay? We could imagine, for example, that a topoisomerase could clip one strand, and when it does, that tension that was in there will be released because that strand will just start flipping around the other one and take care of itself, right? Well, this particular topoisomerase is called DNA gyrase. DNA gyrase is a type of topoisomerase. It's a specific example of a topoisomerase. And it does something even more remarkable. Instead of clipping one strand, leaving one strand intact and the other to unwind around it, it clips both strands. And it clips both strands and allows that unwinding to occur, and then it puts both strands back together. Now, that may sound trivial to you, but it's not. Because that means it's got to be holding on to one side as it allows the other side to unwind. Because if it doesn't hold on to this side, it can't put the two pieces back together after it's taken them apart. Okay? Topoisomerases come in two forms. One form is the first one I described to you. It clips one strand and allows the unwinding to occur. That's called a type 1 topoisomerase. 1 meaning it clips one strand. DNA gyrase is an example of something we call a type 2 topoisomerase because it clips both strands, hangs on to the end, lets it do its thing, and then puts everybody back together. So type 1s clip 1 strand, type 2s clip 2 strands. Pretty remarkable. Now, topoisomerases have a variety of functions in cells. This one in DNA replication is a very important one. There are other functions that topoisomerases have that I won't have time to talk about here, but if you're curious, come and see me. Okay, so we've covered two important enzymes at the replication fork. We've covered the helicase, we've covered the topoisomerase. Okay? I've already told you about the fact that there's an enzyme that makes primers. It's called primase, P-R-I-M-A-S-E. And primase, which is shown right here, okay, is looking for places to put down a short stretch of RNA because it's putting that primer down necessary for DNA polymerase to elongate. For the leading strand, it only had to put down one, and then that the strand got made from that. For the lagging strand, every few nucleotides, a new primer has to be put down. So the primase on the lagging strand is a very busy enzyme. It's a very busy enzyme. It's got to be continually making primers so the DNA polymerase has something to elongate. Another thing you see here called SSB is also important. SSB stands for single strand binding protein. Single strand binding protein. Why does the cell care about something, as its name suggests? Of course, it's binding to a single strand. Why does the cell want something to bind to single strands? Well, it's at this point that the cell is in its most jeopardized condition. If something happens to that strand, the cell's hosed. Because the strand goes flying away, you've broken the chromosome, the cell doesn't have a way of replicating this strand because it's already replicating this one over here, and that gets thrown away. So big problem. Cells therefore protect single strands as much as they can with single strand binding protein. So they're there for protection. Turns out they also interact with the DNA polymerase 3 and help the replication to proceed efficiently. Okay. Now, there's two other things that I want to talk about on this at this replication fork, and they're a little confusing. They're a little confusing. One isn't so confusing. I'll show you a figure in just a second, but it's Dna Ligase. Okay? Dna Ligase. The other is that there's a second DNA polymerase at near the replication fork, and that second DNA polymerase has a name of Dna Polymerase 1. It's called 1 because it was the first one discovered. 3, of course, was the third one discovered. Okay? Now, why does a cell need an additional DNA polymerase over here, and why does it need an enzyme called Dna Ligase? Okay? Well, let me save that for a minute while I talk about a third protein, and then I'll turn attention to that. The third protein I want to talk about is this guy here called the sliding clamp. This is the last one we'll talk about at the replication fork. The sliding clamp. It's also called the beta clamp. Okay? In fact, it's most commonly called the beta clamp. There's a similar protein in eukaryotic cells, and it's called pCNA. Okay, for what that's worth. pCNA. They all have the same function. The sliding clamp, if you think, what is a sliding clamp? Well, it slides, and it is a clamp, right? That's where the name comes from. What does it do? Well, the sliding clamp, as you look at it, looks like that. And if we look at my fingers here, a sliding clamp looks like this, right? It can open, and it can close. And it can open, and it can close. What does a sliding clamp do? A sliding clamp is there to hang on to DNA on one side, and on the other side, it hangs onto DNA polymerase 3. It hangs on to DNA polymerase 3. So what is the effect of the sliding clamp? The effect of the sliding clamp is to hold the DNA polymerase 3 onto the DNA and keep it from flying away. It's because of this that a DNA polymerase 3, once it gets started replicating a DNA molecule, generally will not come off for a long time. It will not come off for a long time. Okay? It's what we call very processive. processive. Okay? A polymerase or an enzyme that gets onto DNA and stays on there is processive. It proceeds without jumping off. By contrast, DNA polymerase 1 is not processive. DNA polymerase 1 is what we call progressive. It progresses from one place of replication to another very, very readily. It only stays on for a few nucleotides, under 100, and then it goes flying off. Why does it go flying off? Well, nothing holding it. DNA polymerase 1 is not held to the DNA by a beta clamp. So the beta clamp ensures processivity. Okay? It ensures processivity. No beta clamp ensures progressivity. And we'll see that that's actually important for DNA polymerase 1 as I get talking about that. Questions about that? Okay. Then, let's take a look at what is actually happening here. Starting, that shows you the reaction, okay? Remember I said the polymerase is catalyzing the joining of an incoming nucleotide onto the 3' end of an existing one, and you can see the chemistry that's going on there, the attack of the phosphate by the oxygen of the hydroxyl, okay, the loss of a pyrophosphate, which is shown right here, and the formation of a phosphodiester bond. That's the polymerization reaction catalyzed by DNA polymerase, okay? Now I want to talk about the DNA polymerase 1 and the DNA ligase, both of which we find in the same general location. In order to get you oriented for that, I need to remind you about the nature of the lagging strand. The lagging strand, of course, has Okazaki fragments. And I want you, and by the way, for those of you who've come to my office, you'll know I'm a very big believer in visualization, okay? I like for you to visualize and be able to visualize, and I'm going to take you through a little visualization exercise here. If I were to visualize a leading strand, I would say right here at the beginning I have a short segment of RNA. Attached to that is DNA, and it just stays DNA all the way, all the way, all the way. If this were linear, it would just continue along like this. If it were circular, what would happen? Well, the bacterial one is circular, so I have RNA, I have DNA, 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 oh, and now I've hit RNA. You vision that? Okay. Now, what happens when the polymerase gets to the RNA? If we simply join the ends together, we'll have a molecule that's part RNA and part DNA. No good, right? It's got to be all DNA. That means that the RNA that we've got here has got to be removed. How does it get removed? Well, let's think about this. We are going, at the very far end of the RNA, we're talking about the 5' end of the RNA, because that's where everything started. We're going 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', 3', and now 3' is right next to the 5' RNA. Right? The DNA polymerase 3 can't touch that RNA. It can't do anything with it. If nothing is done, then that will go, the cell will have a problem. The RNA polymerase 3 at that point goes flying away. The beta clamp lets it go, it gets out of there, and now there's this gap. There's this 3' end, and there's this 5' RNA, and there's a gap, there's a hole in between them. This is where DNA polymerase 1 comes in. DNA polymerase 1 comes in, and DNA polymerase 1, in addition to having a 5' to 3' exonuclease, and a 3' to 5' proofreading 5' to 3' polymerase, and a 3' to 5' proofreading exonuclease, DNA polymerase 1 has a third catalytic activity. It's another exonuclease. But it's not a 3' to 5' like the proofreading one. It's It's a 5' to 3' one. And that makes perfect sense. Because what's the end of the RNA? That's a 5' end. And that 5' end, if the 5' to 3' exonuclease sees it, it grabs it and it starts chewing it away. So the function of DNA polymerase I at a replicator near a replication fork is to remove primers. A second function of DNA polymerase I at the replication fork is as it's removing primers in front of it, in the back of it, the 5' to 3' polymerase is filling in the gap with DNA. So in essence, the RNA is being replaced by DNA. That's pretty cool. That function is essential for E. coli. Okay? If that function is lost, the E. coli cell dies. Okay? What's the end product of that? Well, the end product of that may not seem to you like it's a little hard to visualize, but as the polymerase is filling in with DNA, where the RNA was, okay, it goes chewing, 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 it actually leaves a tiny gap between what now is DNA in front of it and what is DNA that it has left behind. It's what we call a gap. It means that there's one phosphodiester bond that is missing to completely close the circle. One phosphodiester bond that's necessary to join the 3' end of this guy with the 5' end of this guy. Everybody envision that? Okay. This is what DNA ligase does. DNA ligase joins strands that are separated by a single phosphodiester bond. It uses ATP to do that, and that's the function of DNA ligase. Now, I've just described this process occurring for the leading strand, which only needs to do it really once, right? The lagging strand has to do exactly the same thing, but it has to do it about every 100 or so nucleotides. DNA polymerase has to come in, it has to remove that primer, it has to fill it in with DNA, and DNA ligase has to join those things together in a phosphodiester bond. That's the function carried out by DNA ligase. That's the function carried out by DNA polymerase. Questions about all that? Yes? Yes, the question is, are there 3 catalytic activities on DNA polymerase I? The answer is yes. First is a 5' to 3' DNA polymerization activity. That's what makes the phosphodiester bonds. Second, there's a 3' to 5' exonuclease activity. That's what proofreads to make sure it's gotten as accurate as it can. And third, it has a 5' to 3' exonuclease for removing RNA primers. Yes? What's the point of having both kinds of polymerase? That's a really good question. Okay? We don't really know why cells have so many specialized kinds of polymerase. There's also a DNA polymerase II that's found in E. coli. And polymerase II appears to function primarily in repair of damaged DNA. But why doesn't the regular polymerase do that? Because the unfortunate answer is that's the way it evolved. If we look at our cells, we've got about 10 DNA polymerases with some very specialized functions. And I can't tell you, and we don't completely know why there are so many or why the systems have evolved that way. Yeah. Yeah?
Does the ligase also close the exonuclease?
So the question is, does the ligase also close the loop that the exonuclease causes? Well, remember that the exonuclease is basically moving that gap. Right? Because it's moving it forwards, but the polymerase is filling in behind it. So as long as you have 2 pieces that are separated by one missing phosphodiester bond, that's the only thing that DNA ligase will work on. If I had a bigger gap, I'd have to fill it in first. Okay? Okay. So that's what's happening at a replication fork. It's pretty remarkable that this happens as rapidly as it does. I want to say a word about topoisomerases. Topoisomerases do incredible things. You've already seen how they keep strands from getting untangled by relieving tension. They can also create tension. Okay? They can create tension, and you can actually see this in a circular DNA very readily. Okay? I told you that the B form of DNA has about 10.5 base pairs per turn. If I have a circular DNA with 10.5 base pairs per turn, I have something called a relaxed circle. And it's just like its name would suggest. It's just lying there, no tension, no anything. Imagine I take one of those strands and I wind it around a couple times, and then I join it back to the other side of the strand. I will have created tension. I can create tension by adding more twists. I can create the same tension by removing a couple twists. They both have the same effect. Okay? If I do that, I create something called supercoiling, because the tension causes the DNA to wrap upon itself. Instead of just lying there relaxed, as you see here, it has wrapped upon itself. And wrapping upon itself is a very common thing for supercoiled DNA to do. It has a couple of effects. One of the effects it has is it actually comes as a physically smaller entity. Fitting DNAs into cells, we've already talked about some of the logistical concerns about that, and supercoiling helps a little bit with that process. Supercoiling, as we will see later, also helps to create tension to separate strands. And that may be a factor in transcription, the synthesis of RNA that we'll talk about after I finish talking about DNA. When we talk about supercoiling, there are some terms and some simple equations that people use that I want to show you. Let's see, before I do that, yeah, here we go. We can see schematically here what I've just described to you. I've got something that's 10.5 base pairs per turn. I peel this apart and I create some tension and so forth by either putting in twists or removing them, and I see that this guy adjusts by supercoiling, forming a superhelical structure to supercoil, and that removing of those things can be catalyzed by topoisomerases. So a topoisomerase can not only relieve tension, it can also create tension. Okay? Now, the DNA replication fork, it's not creating tension. It's definitely relieving tension. Okay? Was that your question, or was there another question?
The slide says superhelix formation relieves tension. Relieving tension by supercoiling.
It does. Supercoiling actually relieves tension, and that's a little bit more involved than I want to get into here. But to answer your question, yes, it does. Because by forming these superhelical structures, what it's able to do is to go back and be 10.5 base pairs per turn. It takes super twisting to be able to do that, and that's why supercoils actually form. Okay? But yes, it does do that. It relieves the tension. So even though this looks like it's very tense, the actual strands are not tense because they have gotten back into 10.5. Okay? All right. Well, there's, as I said, a simple equation that we use to describe that. I'm going to show you that simple equation, and that's right here. The simple equation says L equals T plus W. L is a linking number. I'm going to talk about that in a second. Okay? Twists, I think we can understand pretty readily. It's the number of times two strands wind across each other. We have 10.5 twists per turn of DNA. Okay? Not per turn. 10.5 base pairs per turn. That turn of DNA is one twist. I'm throwing out the wrong terms. Okay? 10.5 base pairs per turn. That turn is a twist. What's a writhe? A writhe is whenever we see a superhelical structure form. This guy here has two writhes. You see that the double helix crosses itself twice. A writhe can be positive. A writhe can be negative, and there's a convention whether it goes clockwise or counterclockwise, and we're not going to care. Okay? But writhes count the number of times the double helix crosses over itself. This happens with closed circular DNA most easily for us to visualize. Okay? It does happen with linears, but there are other considerations with that we won't talk about. So what's the linking number? The linking number is simply the sum of the twists and the writhes. When we have supercoiling, the linking number will not equal the number of twists because there are writhes. Whenever there are positive or negative writhes, there is supercoiling going on. Whenever there are positive or negative writhes, there is supercoiling going on. Okay? I'm not going to ask you to recognize which way it goes. I'm not going to ask you to make structures like that. But I do want you to recognize this equation and what it tells you. It simply tells you if supercoiling is occurring, because if W is not equal to 0, you know that writhing is occurring. Okay. How much doing on time? 10 minutes. Okay. Questions on that? It's Friday, isn't it? You guys want to hear a joke? Friday, right? My Crunchbird joke. Okay. This is a joke a student told me. It's one of my favorite jokes also. Crunchbird. Lady goes looking for a birthday present for her husband. She goes to the local pet store and tells the pet store owner, she says, I want something really different for my husband's birthday this year. Okay? Pet store owner says, no problem. He goes out, he comes back, and he's got this iguana, the great big iguana, on a leash and hands it to her. And she says, I don't think I want to clean up messes of that around the house, you know, and so forth. And he says, okay. He goes back and he comes back and he's got this long python. And he drags it out and shows her, and she says, I don't think I want that in the house, you know. This goes on for several weird organisms that he's got, and every one he brings out, there's some problem. And he's getting a little exasperated, and he says, I know what you want, lady. He says, you want a crunchbird. And she says, what's a crunchbird? He says, you see that little bird on that perch over there? And she looks across the room, and there's this little tiny bird sitting on this perch. She says, yeah, and he says, watch this. And he says, crunchbird, chair. And the bird jumps off of that perch and it flies over to this chair and it demolishes the chair right in front of her eyes. She said, that's amazing. He said, you ain't seen nothing. Crunchbird, desk, and there's this great big wooden desk in the corner. And the bird flies over to this desk and it's sawdust in a couple of minutes. She said, I gotta have that. That's incredible. She grabs the bird. How many people have heard this joke? Nobody's heard. Okay, well, a couple, all right. She grabs the bird, she puts it on her finger, and she brings it home, and she walks in, and she says, honey, I got something for your birthday. He's reading the paper, and he looks up, and he says, what is it? She says, it's a crunch bird. He says, crunch bird my ass. Okay. Oh, everybody's pained at that one, right? You know what the snail got arrested for, right? Assault? Okay. You know why the guy got fired from the orange juice factory, right? Couldn't concentrate? Oh, okay. Enough pain. All right, we'll get back to some other pain here. Let's talk about a cool application of our knowledge of DNA polymerization, okay, that's used by laboratories everywhere in the world, okay? This was a technology that was invented by a man named Kary Mullis back in the mid-1980s as he was driving home from his laboratory in Southern California. And some people will call this genius. I will not call this genius, although it is surprising that others had not thought of it before him. Okay? Basically, Kary Mullis figured out how to do in a test tube what cells do inside of them and do it incredibly well. He figured out how to efficiently and specifically replicate a desired stretch of DNA and no other. Okay? His invention is called the polymerase chain reaction, or PCR. Okay? And PCR, I'm going to show you schematically, operates as follows. What PCR allows a person to do is, as I said, replicate a specific stretch of DNA. And with the technique of PCR, one can start with a single copy of a DNA sequence, and over the span of 30 cycles, which you can do in a couple of hours easily, theoretically produce over 1 billion copies of that. Okay? So as a consequence, if you have trace amounts of DNA left at a crime scene, or you have trace amounts of DNA that are found at an archaeological dig, or you have any particular gene that you want to isolate or whatever, you can amplify specifically the thing that you're interested in, and in a couple of hours actually have it to work with, which is remarkable. How does it work? Let's imagine we've got a DNA that contains a sequence that we want to amplify. I've got that DNA shown here. What I don't have shown here are all the other pieces of that DNA from that cell that I don't want to amplify. So I want you to remember those as I'm talking about this. I've got millions of other pieces that don't contain the sequence that I'm interested in. I'm only going to follow the one that has the sequence that I'm interested in here. Everybody with me? If I want to do this, if I want to use this technique, what do? Well, the first thing I have to do is I have to separate the strands, because remember, DNA polymerase wants to have those strands separated, and I'm not in a cell, so I don't have helicase. Boiling will separate strands without damaging the DNA. The strands will come apart. And it works because what holds strands together? Hydrogen bonds. And how do you break hydrogen bonds? With heat. Boiling separates the strands. That's the first step in the process. Second step in the process involves addition of what we call primers. Oh yeah, DNA polymerase needs primers. Well, if I put RNA primers on there, I've got a problem, because I'm going to need to replicate this thing eventually, and I can't copy the RNA. So instead of using RNA primers, I use DNA primers. Where do I get DNA primers? I go to my laboratory machine and I tell it, make the following sequence of DNA. And the machine will chemically synthesize those little primers. They're typically about 20 nucleotides in size. And they have a specific sequence. The specific sequence they have is one of the sequences is complementary to this section, this left section over here. The other is complementary to this over here, so when the two were together, they were basically flanking this region in the middle that I was interested in. So I could make primers that specifically annealed only to regions around the sequence of interest. They will not form duplexes with all the other millions of pieces I've got out there, because these sequences don't appear in those. Okay? So the first thing I've done is I've told the polymerase, here is where you're going to replicate. And all of these other things that are out here won't have primers because there's nothing complementary to them out there, and the polymerase will leave them alone. The primers, if there was genius, that was where the genius of this technique was. Okay? The DNA polymerase is added, and 4 DNTPs was added, and no surprise, if you have a prime sequence, replication is going to occur here, but all these millions of others that are out here will not have any replication occur. Okay? And then, all I do is repeat what I just did. Boil, anneal, replicate. Boil, anneal, replicate. Each of those takes place at a specific temperature. And to make it really simple, they realized that most enzymes don't survive boiling. So they went out and they found a DNA polymerase in an organism that lives in Yellowstone's Old Faithful geyser that did tolerate that temperature, and so they didn't have to add enzyme every time. They just had to boil, anneal, replicate. Boil, anneal, replicate. Every time you do that, you theoretically double the number of strands that are there, so that after 30 cycles, you theoretically have over 1 billion strands specifically of the thing that you're after. I saw a hand over here.
Yeah, what does the replication
DNA polymerase. It's called TAQ, T-A-Q, DNA polymerase, because TAQ stands for Thermus aquaticus. You don't need to know that. But that's the name of the organism that lived in that hot spring. It's added by someone before the reaction starts. Okay, I'm running over just a little bit, so I've got to squeeze in our song, which is about DNA, and it's a very energetic one. I want you to join me on this one. It's called BDNA. Phosphates are in nucleotides, I say phosphates. Cover bases inside, I say phosphates. Span the 5 and 3 primes, there's no need to be real mixed up bases. Carry info you see, I say bases. Are all complementary, I say bases. Like A, T, G, and C, they have got to be all paired up. Boom, 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 boom. It's fun to play with some BDNA. It's got a boatload of GCTA. I can't hear you. It's got everything a polymerase needs when you meld all the A's and T's. BDNA, it's got a boatload of GCTA. You can make RNAs with a polymerase just by pairing up U's with A's. Proteins full of amino A's, I say proteins come from mRNAs, I say proteins require tRNAs. There is more you need to translate codons. Like our friend UAC, I say codons come in clusters of 3, I say codons have one base wobbly. Now you can go forth and translate. Boom, 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 boom. It's fun to play with some BDNA. It's got a boatload of GCTA. With those hydrogen B's and right-hand helices, anti-parallel 5s and 3s. Okay, that's enough. See you guys on